So our next session we entitled Lessons from Cell and Gene Therapy for Other Conditions. And here Keith and I thought it might actually be nice to hear a little bit what folks do for other diseases in the cell and gene therapy space, especially, of course, for cancer. And for that, I'm very happy to have Aude Chapuis here, Dr. Chapuis from the Hutch and the UW, and she's going to talk to us about optimizing T-cell receptor gene therapy for cancer. Thanks so much for coming, Aude.
Thank you very much for inviting me. Okay, so So I actually started to work on HIV way back when, and it's funny that actually things turn around. We actually infused T cells that we had raised against HIV-specific antigens in patients in a first in human trial. We found a lot of things. We couldn't stop the immunosuppression, but it was an interesting experience. So it's actually nice to see that things are coming back. So since then, I really changed focus, and and started to look at T cell therapy for cancer. And so essentially, everybody knows how a cytotoxic T cell work. It recognizes with the signal 1, which is really the specificity. And on the T cell, it's a lock and key phenomenon. It's extremely specific. And the idea is that the T cell would kill the tumor cell, the virus-infected cell. That dies off, and then the T cell goes on to serial killing. There's a signal 2, here characterized very simplistically by PD-1. If PD-1 is expressed on the T cell, it can bind to PD-L1, and the signal is turned off. And that, you'll see, has implications for Merkel cell polyomavirus that we have been targeting. So anyways, with T cell, you can start to target a lot of things in tumors, tumor-specific antigens, overexpressed self-antigen, and also viral antigens. What I'm going to be focusing on today is the Merkel cell polyomavirus that we have a full program on in the lab. Merkel cell carcinoma, aggressive skin cancer, even worse than melanoma. It's rare. However, the incidence is rising because the baby boomers are coming of age, and they have been exposed to a lot of sun, which is a risk factor for Merkel cell. And interestingly, 80% of these Merkel cell polyomavirus, Merkel cell carcinomas are caused by the Merkel cell polyomavirus. So they have basically an integrated virus in them. So it requires a mutated form, so there has to be some events before the truncated, non-replicative Merkel cell polyomavirus can insert itself into the DNA of the cell and cause the cancer. So it encodes the T antigen protein, which targets several tumor suppressor proteins. It's persistently expressed. It's necessary for the survival and the proliferation of the Merkel cell cancer. And that obviously is a fantastic target for an immunologist, which is basically a target which is non-self, and that you can raise T cells against. Merkel cell is responsive to anti-PD-1 therapy in 50% of cases. However, as you can sort of see, there's a plateau that forms, although the data is still not enough to be able to see the trend long term. I think the message here is really, there's still 50% of patients who still need some form of therapy. As many tumors driven by viruses, the Merkel cell polyomavirus has the tendency to decrease the amount of HLA expressed on the tumor. However, this is reversible, and you can either do this by giving interferon gamma in the tumor microenvironment, either directly or systemically, or you can also do this by one shot of radiation. For example, 8-gray radiation will start to increase the HLA, and obviously that for an immunologist we want to make sure that our antigen is actually presented on the surface so that our T cells can see it. We started when I was a postdoc. I was a postdoc in Phil's Greenberg's lab and then Cassini's lab, and we did a lot of endogenous adoptive T-cell therapy. We targeted HIV, I talked about, but melanoma, leukemia, and also Merkel cell. And the principle here is really to take a leukapheresis from the patient and then identify, by stimulating with peptide pulsed autologous dendritic cells, T cells that are in the patient, and then expanding those, selecting them by single cell sorting, expanding them some more, and then giving them back to the patient. And that's exactly what we did for Merkel, except that we had a little bit of goodies that were to specifically address the characteristics of this disease. We first did radiation to one tumor site, so not even if the patients had multiple tumors, we would just irradiate one with 8 gray for HLA upregulation. And then we combined adoptive T-cell therapy, which was only CD8 T cells to start with a checkpoint inhibitor, which was pembrolizumab in this case. So we had nice responses, or at least encouraging data, where we had 4 of the 8 patients who achieved either a PR or a complete remission. 2 of them are still alive and well. Some of them have progressed after a time of initial response. So if we look at the results of this time, for example, this is a lady who had a big blob in her neck. The blob was irradiated, and we also saw that actually with time, both the non-irradiated and the irradiated lesions decreased. We saw persistence in this trial. We were also able to follow our T cells with their specific CDR3. So basically, the products that we are infusing are polyclonal with Merkel cell polyomavirus specific T cells, and you can follow those now either in the blood or at the tumor site. And in patients who were responding, we really saw that there was a preferential accumulation of these clonotypes that were present in the infusion product at the site of the Merkel cell carcinoma, and that when you did immunohistochemistry, you could also see now a nice infiltration of the tumor cell, which was not present before. Another aspect that we saw in this trial was what we call this epitope spreading, so basically new responses to non-targeted epitopes. In this particular case, in green, we're targeting the common T. There's 2 splice variants for the Merkel, and we saw that there were new CD4 T cells responses that were not present before, and that was after the transfer of T cells. And we were also able to increase the CD8 T cells by about 500-fold at the site after even just really quickly after 14 days. So basically, new responses driven by that. Another indirect measure of efficacy, which I'll put in the category of acquired resistance. So these are the patients who respond initially and then progress. And it turns out that those patients will give us the most information about, how our T cells work and what do we need to be improving. And so I want to focus a little bit on one of those patients. There's a patient who received B35 specific Merkel cell polyomavirus cells. He had quite a bit of a tumor burden at the start. He had already received checkpoint inhibitors, and nothing worked. And so when we gave him the T cells, we saw that about 30% of his total CD8 now were Merkel cell polyomavirus, which was new compared to what he had before. And he initially didn't respond as well as we wanted, so we started to add sort of the kitchen sink, anti-PD-1, anti-CTLA-4, 2 checkpoint inhibitors. And for a period of over a year, this person nearly completely regressed his Merkel cell. All he had was a little spot in his leg. Unfortunately, after a year, the cancer came raging back. And so we started to try to understand that we obviously had a direct cause and effect. We had given the cells, and he had responded, and now he was not responding anymore. So when we looked at the time of initial progression here, we're looking at single-cell RNA sequencing data, so basically transcriptomics on just full PBMC. And we saw this population of activated CD8 T cells that was only present at the time of response in this patient. And when we look at their gene expression compared to other CD8 T cells, we saw that they really had overexpressed genes suggestive of activation, metabolism, and cell division. And indeed, when we looked into this patient at the time of response, so on the top panel we have before T cells. You can see the tumor is a cold tumor. There's no T cells in there. At the time of response, you see the tumor gets infiltrated with many, many T cells, and we were able to show that those were the T cells we had infused. And then at the time of acquired resistance, again, the tumor is cold. So what happened to this tumor? And when we started to look more precisely, so here again we're looking at single-cell RNA sequencing data of this tumor. On the top left-hand side is the tumor before, and on the bottom is the tumor after. You can see that genetically these two tumors coming from the same patient, just at different time points, are really very genetically diverse. And when we started to look at our HLA-A, which was not targeted, you can see that there's expression both before and after. However, when we looked at HLA-B, there was a complete downregulation of HLA-B at the time of progression, and that was the HLA that the TCR was targeting. So basically, the tumor was able to downregulate the targeted HLA-B in order to escape the T cells, and we actually were able to see that in 2 patients. In one patient who we were targeting HLA-A, so basically exactly the reverse. So this was very specific of a tumor mutation to or a tumor adaptation to under the pressure of the T cells. So we were happy to see that our transfer cells were clinically active. However, we identified quite a number of challenges through the trial. The non-gene modified T cells product rely on the patient's endogenous T cell repertoire. So basically, whatever we can raise against the virus is going to be what is already present, maybe at lower quantities, but it's already present in the virus, in the patient. And we know that in patients who have progressive disease, if we take the T cell clones, the CD8 T cell clone, we look at how their EC50, so how they react to produce interferon to the target, we can see that in the patients who have progressive disease, who have bulky disease, the T cells that are in the patients are just not very good. Their EC50 is high, meaning that they really don't have a lot of reactivity to the targeted antigen. Probably, possibly by activation-induced cell death. The other issue with endogenous T cell therapy is that we have suboptimal expansion of these cells, probably also because they're exhausted. So now you're taking them out of the patient, you're trying to stimulate them in ex vivo, and it doesn't really work very well. And it takes quite a long time for these patients. They progress extremely rapidly, so we need something with a faster turnaround. So we decided to embark on adoptive T cell therapy using genetically modified T cells and basically identify a high affinity TCR against Merkel cell polyomavirus, then we have fixed affinity, high affinity that we can then plug into our selected substrate cells, so selecting cells which would persist better. So how do we identify high affinity TCRs these days? There's quite a bit of methods. Groups have looked into generate high avidity T cells and immunize HLA transgenic mice. Unfortunately, that has caused issues because the mice don't have the human genes that are presented by the thymus. So what you're getting in these mice are TCRs that are of extremely high affinity and that can now recognize other peptides and cause serious damage. You can tweak the amino acid residues contacting the HLA peptide complex. However, there again, you have to be very careful because if you touch the CDR3 or a certain zone of the CDR3 or CDR2, you can start to also increase the promiscuity of these cells. So basically, these TCRs now recognize not the peptide, but the HLA, and also cause a lot of damage, including patient death. We decided to go another route to identify high-affinity TCRs from the peripheral repertoire of normal donors. So the idea is to screen normal donors and to try to find rare high-affinity T cells specific for self-antigens, but we actually extended this method now to the Merkel cell polyomavirus that are hiding in sort of in the peripheral repertoire that are really high affinity, so they have killing ability, but that they don't cross that threshold to be toxic. How do we do that? We basically take HLA-matched donors, many donors. We've gone from 15 to 20 donors, and we generate T cell lines. So you have to be able to get to know the peptide and the HLA restriction that you're targeting before you start. And then we serially sort these cell lines. We pull them, we serially sort them with the idea that we're only going to take the ones that are the stickiest to the tetramer. So doing that, what you can do is a ratio, essentially looking for the most highly enriched in the sorted tetramer high population versus the baseline. And doing a ratio, what it does is that it allows for, so you don't really care anymore about the frequency in your sorted population, but it's really you're looking for TCRs or clonotypes that increase their ratio with decreasing concentration of tetramer. And indeed, we now shown that small differences in tetramer binding in the EC50 here shown here in the functional avidity of these cells translate into bigger differences in the killing ability. So we really need high affinity T cells. This method now we've been using for a number of different targets, including viruses. And for the Merkel cell polyomavirus, we were able to show since we were going to the clinic, we had to find a high affinity T cell to translate, and we had three options. In black, we saw what we call our straw man, which is a TCR of highest avidity clone isolated from a patient, so with standard methods. In blue, a TCR isolated from the patient who experienced a CR after endogenous T cells, and we were able to show that the CR was mediated in part by the TCR that we isolated, and in red, isolated from the matched donor using our high throughput method. You can quickly see that the black one doesn't really do well, and it was sort of a neck-to-neck between the patient one and the normal donor one, and we actually ended up taking the method one, so coming from a normal donor, because it could better recognize a variant of the peptide that we were targeting that is a normal variant in the Merkel. So there was higher affinity and therefore promiscuity, a little bit more promiscuity of that TCR. One last thing I want to touch upon is that, we've talked about CD8 T cells, and our endogenous T cell therapy was done with CD8s, but CD4s are also, as you all know, are extremely important. We want to engage them. And the idea is really to engage them with a class 1 so they can both be brought to the target and help each other, both the CD8 and the CD4. And we were lucky to find out that our clinical candidate, actually, when you do put it in a CD4, it can kill a cell line that expresses the Merkel cell polyomavirus, meaning that TCR is of high enough affinity that it doesn't need the CD8 binding in order to cause a trigger and a killing ability of the cells it's plugged in. Right now, we've treated our first patient. This is my postdoc, Kelly Paulson, named it ATTACK-MCC for autologous transgenic T cells, avelumab, and class 1 MHC upregulation. And now this is basically 2 infusion combined, again, based on the previous trial with radiation and avelumab, and it's ongoing, so no results yet. It's too early. Thank you very much. I think I said I put pictures of all the people that were there that did the job, but essentially these are my source of funding and the people involved. Thank you very much.
Thanks, Aude. Any questions?
When you put this transgenic TCR into the CD4s, do you know if it's recognizing the peptide in the context of class 1 or class 2?
It's class 1.
Okay.
For sure, yes.
Cool.
Yeah, because it's a class 1 TCR, I can't recognize a class 2. It's really very specific. So, it's this tripod thing, right? There's the HLA and then the TCR and the CD8, and you need that stability. So if the TCR is of high enough affinity, you can actually cause that stability, but it has to be of high enough affinity. We have never seen that, for example, in self-antigens.
Maybe one more question if there's. So any interest in coming back to the HIV field again?
It's too complicated.
Thank you.